So I'm going to tell you today about Vaccinia virus and what the virus does to the actin cytoskeleton and microtubules, but already from the talks this morning you've heard how various different pathogens, where is it, Shigella, Listeria, can make actin tails, but there's a number of other bacterial pathogens that also stimulate actin polymerization to help their spread. But this is our favorite bug, which is Vaccinia virus. So the green dots are virus particles, and you see them on actin tails projecting from the cell. And if you have virus strains that can't make actin tails, the virus is less virulent inside an animal model, and it's deficient in its cell-to-cell spread. This is about 8 hours post-infection. So what is Vaccinia virus? Well, for the older people in the audience, it was the vaccine we were given to protect us against smallpox the last century, which killed about 200 million people. This is why George Bush is worried about bioterrorism with smallpox. It's a very large virus, encodes a large number of proteins. We only know the functional localization of about half of them, and the virus undergoes a very complex replication and morphogenesis actually at the microtubule organizing center of an infected cell in the cytoplasm, and it actually encodes much of its own replication and transcriptional machinery, and it assembles several different forms of the virus. The first form of the virus, I don't have time to show you today, moves on microtubules. It moves towards the TGN into which the virus puts its own proteins. This envelops this virus particle to form an IEV, which moves out on microtubules. This fuses with the membrane. We don't know what's going on here. And it's the virus on the outside that makes the actin tails on the inside. I'll explain how it does this. Now, this is a nice schematic from a textbook or a paper, but this is actually what it looks like in real life. So this is the DNA in the nucleus. This is the viral factory where the virus assembles its own DNA, all in the space of 8 hours. This is a marker on the IMV, the first form of the virus. A proportion of these wrap at the TGN to form the second form of the virus, the IEV. And basically what you can see, this is only 3 minutes, but the virus is a little brick. It's 250 by about 300 nanometers, and you can see there's a lot of movements going on. So clearly these movements are not going to happen by diffusion. They have to be an active process involving the cytoskeleton and various motors. Now I'm not going to show you anything of this one, but this shows you the IEV, these white dots. This is a spinning disc confocal we took many years ago, of the IEVs moving out to the cell periphery, hopping from microtubule to microtubule towards the cell periphery. Again, they move about 1 micron per second, although you will see faster velocities of 5 microns per second. And we know that the virus actually recruits conventional kinesin, so kinesin 1. So we can image kinesin on stationary particles and moving particles. It's still not clear exactly how the virus recruits that kinesin. There's some suggestions of molecules involved, but there's obviously a big complex on the virus surface that recruits the motor. In terms of the actin, where most of our research has been aimed, it's been easier because the tails move a lot slower, so they move in a very precessive fashion, about 0.2 microns per second. And you'll see 2 virus particles that come up here. It will repeat itself on microtubules, and then one of them here will fuse and nucleate an actin tail. So the question is, how is the virus that's on the outside able to nucleate an actin tail on the inside? And what we know from our, just to show you for the skeptics in the audience that the actin tails really are only at the plasma membrane, this is a confocal slice at the very bottom of the cell. So the actin tails are on the inside, the virus is on the outside, so you're looking down on the cell, and the viruses are trapped between the cover slip and the plasma membrane, and the actin tails inside, and this is the Z section of the same cell going up through. This is the cytoplasm. You can see there are no actin tails. Everything is restricted to the plasma membrane. So how does the virus do this? Well, once it fuses, one of the membranes that wraps the virus gets left in the plasma membrane, and embedded in this basically viral membrane into which there's viral proteins, there's a protein called A36R. Doesn't look like anything. You can do all the database searches you like, and nothing really leaps out at you of any predicted structures or domains. But what we know is the virus on the outside is somehow able to locally activate Src and Abl family kinases The way in which it does it, we have absolutely no idea. These kinases then phosphorylate 2 tyrosine residues, and this is just showing you the phospho blot. So this is, if you remove the 2 tyrosines, you still see activation of Src. We see Src associated with tails. And when these 2 tyrosines are phosphorylated, the virus recruits a downstream signaling complex that consists of Nic and Grab2 adapters, which link into WIP, wasp interacting protein, NWasp, and NWasp now locally activates up to 3 complex to stimulate your actin polymerization. So if you think about this, this looks like a receptor at the plasma membrane that gets phosphorylated by a Src family kinase, and it recruits a downstream signaling network, very much the same as goes on in many signaling cascades in cells normally. The advantage in this system is it's very localized, so it's very easy for us to study with dominant negatives and RNAi, and you can see this is recruitment of the endogenous proteins. It's very easy to see. I should say, we know if you get rid of this tyrosine, the whole system works. You get actin tails, but you get less of them. It's less efficient, so the Grab2 seems to stabilize the network. So if you're interested in a signaling network, there are many questions that you could ask, but I guess the main ones are, you'd like to know all the components in the network, and I've drawn some of them here. I'm sure there's going to be other components we've not identified yet. You'd like to understand all the molecular interactions. So these arrows, we'd actually like to know where those interactions occur. We'd like to know structural information about the network. You'd also like to know the dynamics of the complex and subcomplexes. For example, does the whole complex turn over, or does it just sit there and NWasp just repeatedly activates up to 3 and nucleates more actin? So over the last 2 years, a graduate student who's just left in the lab started to address this question: what's going on with the dynamics? So what she did was made a bunch of stable cell lines, and we've done this in HeLa, where we've expressed GFP tagged all the components that were in that schematic, and you can see they're nicely recruited to the actin tails. She's also rescued the Nic nulls and the NWasp nulls with GFP Nic and GFP NWasp, and she gets identical results. And the idea was to come along, image them, and just bleach virus particles and look at the recovery to see, do they recover, and what are the dynamics. So this shows you a typical sort of bleaching image. At the very beginning, you'll see it dims and it gradually comes back. This is GFP NWasp, and we use an RFP tagged virus so we can know we're looking at a virus particle. And these are the sort of curves that she gets. And immediately it becomes apparent the proteins are not turning over at the same rates. They're all different. And if you look at the half-lives, what you see is Nic and WIP are turning over about 800 milliseconds. NWasp, which is the downstream factor, is turning over about 2.5, 3 times slower. Grab2, which comes in here, is turning over at the limits we can measure on our microscope, 140 milliseconds. That's this one here. So earlier I said the whole system works if you get rid of this tyrosine. If you get rid of this one, there are no actin tails. So Ina came along and she did exactly the same analysis where she gets rid of this tyrosine, because the argument would be NWasp is slower because Grab2 is also linking into the NWasp and stabilizing the complex. So she gets rid of this tyrosine. Now you see the complex still works, all the curves shift, and basically everything speeds up. So now Nic and WIP are turning over about 400 millisecond half-life, but NWasp again is still much, much slower. So immediately this says to us there has to be something else that's stabilizing the NWasp. Now it could be molecules we don't know about, and I suspect there are some. Good candidates would actually be the kinases themselves, where their SH3 domains may link into the proline-rich domain of NWasp. But there's another linkage which you don't think about, which is actually the output of the whole system. The actin itself. NWasp activates the up2-3 complex, but it also binds actin monomers, and it will bind to the ends of actin filaments. So any filament that's nucleated by up2-3 or filaments near NWasp, NWasp can grab hold of these filaments. So to explore this possibility, we took advantage of an observation by Jack Taunton's group in which they've made a mutant which makes mutations in the WH2 domains of NWasp, which mean it can no longer bind actin monomers and it can't interact with the growing ends of actin filaments. So the idea is, with any linkage to the actin downstream, we're getting rid of it. She expressed this protein in NWasp nulls, so we have no endogenous protein because it's rescued. And what you can see is this would be a rescue of a wild type. There's a virus here, there's a nice tail. The RA mutant, you have the virus, you still get actin tails, they're shorter, but you do get them. If you cut off the whole region that binds NWasp, the WA domain, then you get recruitment, that's what the cyan color is, but there's no actin tail because it can't activate the ARP2/3 complex. So Ina came in, she did all the bleaching, and I'm not going to show you the bleaching results, and measures the speed at which the virus is going as she's bleaching it. So the wild type is the normal one that has the 2 tyrosines. The A36Y132F is the one that lacks the GRAB2 site. And basically you can see either for the wild type virus or for this mutant that gets rid of the GRAB2, if we block the connection to actin downstream, everything speeds up. The half-life, NWasp starts turning over faster. And conversely, the virus actually starts moving faster. And there's a nice, perfect, this is all going down nicely and this is going up nicely. So basically what we have is a situation where the faster NWasp is turning over underneath the virus, the faster the virus is moving, which is, we didn't expect. I mean, ultimately, we originally thought all the molecules would turn over together as a complex. The other thing we found is in the WA mutant, where you knock this bit off here, there is no turnover. It basically goes there and the whole system sits there like a brick. This is why the delta WA is such a good dominant negative that people use. I think it's going into all the complexes and it never turns over. If you inhibit actin polymerization as well, there's also no turnover, or very little. So it seems you need actin polymerization to keep the whole complex turning over. So we're now still trying to understand why does the stability of NWasp actually control the rate of motility. And we've talked to a lot of people, and we still don't have a satisfactory answer, and there's a lot more things we still have to do. But one thing you could think about is, I draw it like this. Actually, there's a virus, this is pretty big, it's 200 nanometers across. Actually, there's more than one A36. There's multiple A36s. So you can have cooperativity in the system. So it's not like this, there's one, there's actually lots of them. So you'll have NWasp, which is activating ARP2/3, growing more actin, but you also, any NWasp that's there will be grabbing hold of the ends of growing filaments. So you could imagine a situation where if this is the virus that's being pushed forward, and you have to remember normally there's hundreds of filaments here, some will be growing and pushing, but NWasp will actually be trying to grab the ends of them to stop them growing. If NWasp is turning over faster, you can imagine that it's not going to grab the filaments as easy, and now the filaments will grow for a longer time and push the virus and make it go faster. But now you have a problem. The host proteins in the cell will be trying to block the ends of those filaments as well. So I think we haven't got a satisfactory answer of what's really going on, but I'm sure it's all going to be down to the relationship between NWasp, the dynamics of ARP2/3 and its activation, and how this relates to actin filament elongation and capping. There's some cooperation going on here, some competition between capping, activation, and with other host factors. We'd like to know NWasp. I mean, is it one goes in, activates ARP2/3, and then drops off, or is there multiple cycles? And obviously cooperativity in the system we'd like to understand. And another thing that we're beginning to address now, how much phosphorylation do you need to drive the system? And we've got some tricks where we're actually able to titrate the amounts of phosphorylation on the virus, and we're beginning to see how that affects turnover and how it affects the ability to even make an actin tail. Now I want to leave actin tails there and move on to the last little bit, which is how does the virus deal with the cortical actin? So the virus moves out of microtubules, it recruits conventional kinesin. Something goes on here, it ends up on the outside, signals back inside, I draw it as X, like a receptor. Maybe it's signaling back through lipids. That's probably my best bet. To locally activate Salk-enable family kinases to make your actin tail. But this is a big thing. It's 250 nanometers, and the cortical actin is very dense. So how does the virus actually get through this? Well, I don't have time to show you all the evidence, but we know in the cortex, independently of actin and independently of microtubules, virus particles move. They move about 0.3 microns per second. And we also know that in the cortex, the virus will actually recruit myosin 2B, and we can image myosin 2B on moving particles. So it looks as if a myosin is at least involved, either moving through the cortex, or maybe it's involved in the last step of fusion. We don't know exactly where. The other thing we know is if you need active actin dynamics. If you stabilize the actin, the virus has a hard time getting out. If you get rid of all the actin, it has a hard time getting out. So it needs active actin polymerization to actually get through the cortex and or fuse. It also involves Rho-A signaling. And what we know is the virus actually encodes a protein in its genome that's very conserved. F11L doesn't really look like anything, which is typical of many of the viral proteins. And this shuts down Rho-A signaling during infection. And this is just a quantification of a Lycor Western blot. This would be the uninfected. So this is active Rho. This is total Rho. This is a delta F11L virus. You see Rho levels stay up. But in the normal situation, Rho levels go down. And one consequence of inhibiting Rho signaling is you affect actin organization in the cortex. We're not sure in what way. You affect microtubule dynamics. And all of this contributes to make it easier for the virus to reach the plasma membrane on a microtubule and to get through the actin cortex. So if you get rid of F11L, you actually see the number of virus particles that are released into the media significantly drops. So this is just on HeLa cells in isolation, but if you do a plaque assay, because anything that affects virus getting out will affect its ability to spread. So if you do a plaque assay for 4 days, this is the wild type virus, this is a typical sort of plaque. This shows you the delta F11L. The plaques are much, much smaller, as you can see here. We've counted quite a few. And then we thought one day, well, let's video a plaque. Rather than this, let's see if what we can get out of a plaque. So the hardest part in this is you need a stable microscope. So this is 2 days in the life of the plaque. But the hardest part is actually compressing the movies so you can actually show them in a PowerPoint. But what you can see is we start imaging after about 8 hours when we detect the first infected cell. The virus, this is the wild type on top, virus spreads from cell to cell. There's a sort of wave ahead of the virus front. You lose cell-cell adhesion in the middle, and the virus is nicely spreading. This is delta F11L. There's no loss of cell adhesion. And actually, if you look at individual cells, they're not actually migrating either. And you can see the virus is greatly reduced in its cell-to-cell spread. And if you zoom in, this would be a normal infected cell, the original infected cells here. It's funny, the original infected cell never moves. It always stays where it is and sort of just goes around in a circle. But the other cells, they become infected, they contract, and they begin to migrate away and spread the infection. Now, this is on a confluent monolayer, so what happens in an animal model of infection? Well, if you come along and you spray some virus up the nose of a mouse, what you can do is, this is the WR, this is in the trachea after 1 day. After 1 day it's gone, and you now see it begins to appear in the lungs, and this is the black bars here. This is the virus titer per microgram of tissue. 1 day, 3 days, and 6 days. The black bar, it's in the trachea, it moves to the lungs, then it spreads to the spleen and the liver. That's the wild type. And actually the delta F11L virus, and all this can't do is basically downregulate Rho signaling. Now what you see is it takes longer to appear in the trachea, and basically it has a hard time. You do see it move into the lungs a bit after 3 days, but it doesn't progress. So this virus is very attenuated in vivo in terms of its spread. And we've done this work with Susanna who's in Mariano Esteban's group in Madrid in Spain. But basically, the wild-type virus, the mice, if you give them a lethal dose, they start dying after 3 days, and they're all dead by 8 days. But this virus is completely fine. And I can say it elicits a very nice immune response. So we're interested to know more about how we can use this virus to actually use it for various therapies. And all we've done is affect one protein, which affects Rho signaling. And with that, I will stop and hope I've shown you today that the cortical actin barrier is a spread to infection that all pathogens are going to have to deal with when they go in and when they go out. The virus is recruiting a myosin. Myosin 2's there, but it may not be the only myosin involved, and we're on a hunt for other myosins at the moment. And the virus is manipulating the cortical actin using Rho signaling to M dia. Exactly what it's doing to the cortical actin we don't know, and this is where we have to team up with people like Marek to actually do tomography to look at what is the cortex, what regulates the cortex, and how is it structured normally in non-infected and in infected. And then the last one is to show the lab, and actually the work has been done by, on the FRAP, and the signaling on the virus was done by Ina, who's a graduate student who's just left the lab to go and do a postdoc in Heidelberg, and João, he was a Portuguese graduate student who finished at the same time, and he was working on the Rho A and how inhibition of Rho A affects viral release. And this is the lab and sort of viral projects and non-viral projects we're working on. And thank you for your time, and happy to answer questions.
Can you hear me? Yeah.
Yeah.
So I was wondering in your FRAP experiments how, I was thinking of an alternative hypothesis. Is it possible that what is limiting here is the availability of the free construct? For example, for the NWASP that doesn't bind to actin because perhaps the free concentration is higher, then the turnover rate might be higher as well. In other words, for the construct that are there, for example, I guess what you have is like expressed from exogenous motor.
Right, so we've put them in unstables on lentiviruses into the cells, so the levels of expression are all pretty good. And we actually, it's been a problem, we only bleach the virus particle. We make the smallest region of interest and we chase particles along. So I don't think we're not bleaching a huge area to deplete the surrounding, if I understood correctly.
No, I think the question is whether, for example, if you had to decrease the expression levels of the construct, if there would be a point where perhaps the endogenous interaction would be limiting in a sense that you would not be able to have sufficient free construct to diffuse and to exchange.
Right.
If you see what I'm trying to say.
So you're talking about how much of the GFP compared to the endogenous we have there?
For example, yeah.
Yeah. So all the rates we did in HeLa where we have endogenous backgrounds. We get exactly the same numbers when we do it in the NWASP or Nick nulls, which we've rescued, where all the GFP is only what's there. And we originally made clonal lines of everything and spent ages making them, and over time they all became heterogeneous again. So in the end, we've actually worked with populations of cells, all of which have, varying levels, so it's not like one. So I don't think that's a problem. But we can't explain our observations still. So if anyone has ideas.
Hi, I have a couple of questions. Do you know if the binding to myosin is, how does it bind myosin, and is myosin binding calcium dependent?
At the moment we have no idea how the myosin is bound. It immediately raises questions, right? Because is it in little filaments and polymers, which you'd expect if it's active, but we've never seen anything like that by EM on the virus. And the thing that gets even more intriguing, we don't detect a light chain. We only detect the heavy chain, no light chain.
Do you detect calmodulin?
We haven't looked for calmodulin, that's on the list, and I've suggested it, but yeah, it's a bit of a strange one.
My second question is, in the wave of movement, that looks like integrin mediated. So have you looked at PKA phosphorylation of integrin alpha 4?
All I know is there's lots of alpha integrins and there's lots of betas. We actually haven't done anything with integrins, except for years ago we stayed in some infected cells and there was nothing obvious in terms of changes of localization. But the cell adhesion does change. It goes from calcium sensitive to calcium insensitive. So I assume there is a lot going on with integrins in the system. I need someone who works on integrins to come and tell me what's going on. That's
Mark Ginsberg.
for example.
Hi. Oh, yeah. Very nice talk. Thank you.
Thank you.
So, just to clarify something, is NOS binding the barbed end of the filament, or is binding the side?
It's binding the barbed.
Okay. So
The WH2 domains would, but the ARP2/3 that it's activating, in theory, would bind the side of a filament.
So, I mean, the evidence that I know about is that on G-actin, WA binds G-actin. Do you have evidence that WA binds F-actin?
The WH2 domains in it's either called the WA domain or the VCA domain.
Right.
But the WH2 domains will bind the ends, the free ends, which
right.
mimic a G-actin.
When it's a monomer, but in the filament, I'm not sure it does bind actin.
It will bind the ends of a growing actin filament.
So it doesn't fit into the ARP3 model. So that's you know, so somehow it's an interesting observation, but it doesn't fit into the way we think the ARP3 nucleate the actin.
Right, as you said exactly, it's a model, right? So the question is, ARP2/3 and branching has been shown in vitro, it works very well. It's done at non-physiological concentrations of ARP without its normal regulators. The question is, what's really going on in a cell? And I can say there's work going on now that shows there are very little branches in the leading edge. And we've started the tomography on the tails, and we see practically no branches.
In the tail? Or
in the tail.
Oh, okay.
So I'm not sure, what in the ARP2/3 does in the test tube, I agree, but it doesn't mean that's exactly what it's doing at the leading edge or in a tail.
Okay, thank you.
So there's going to be a lot of interest on this soon, I think.